After studying this module, you shall be able to know about the causes of genetic variation in humans. Human genetic variation. Let us learn about human genetic variation. Human genetic variation is the genetic differences both within and among populations. There may be multiple variants of any given gene in the human population or genes, leading to polymorphism. Many genes are not polymorphic, meaning that only a single allele is present in the population. The gene is then said to be fixed. On average, biochemically, all humans are 99.5% similar. No two humans are genetically identical. Even monozygotic twins who develop from one zygote have infrequent genetic differences due to mutations occurring during development and gene copy number variation. Differences between individuals, even closely related individuals, are the key to techniques such as genetic fingerprinting. Alleles occur at different frequencies in different human populations, with populations that are more geographically and ancestrally remote tending to differ more. Causes of differences between individuals include the exchange of genes during meiosis and various mutational events. There are at least two reasons why genetic variation exists between populations. Natural selection may confer an adaptive advantage to individuals in a specific environment. If an allele provides a competitive advantage, alleles under selection are likely to occur only in those geographic regions where they confer an advantage. The second main cause of genetic variation is due to the high degree of neutrality of most mutations. Most mutations do not appear to have any selective effect or the other on the organism. The main cause is genetic drift. This is the effect of random changes in the gene pool. In humans, founder effect and past small population size, that is increasing the likelihood of genetic drift, may have had an important influence in neutral differences between populations. The theory that humans recently migrated out of Africa supports this. The study of human genetic variation has both evolutionary significance and medical applications. It can help scientists understand ancient human population migrations as well as how different human groups are biologically related to one another. For medicine, study of human genetic variation may be important because some disease-causing alleles occur more often in people from specific geographic regions. New findings show that each human has on average 60 new mutations compared to their parents. Apart from mutations, many genes that may have aided humans in ancient times plague humans today. For example, it is suspected that genes that allow humans to more efficiently process food are those that make people susceptible to obesity and diabetes today.
Let us learn about the genetic variation. Genetic variation occurs due to the difference in the sequence of genes. No two individuals, except for identical twins, are genetically identical. Variation can be seen at every genetic level in DNA, in gene, in chromosome, in enzyme. Now let us learn about the importance of genetic variation. Let us learn why genetic variation is important for us. This is because, in order to better understand ourselves as species and use this knowledge to improve our health. Secondly, it also provides the genetic material for natural selection. Causes of genetic variation. The major cause of genetic variation is mutation. In biology, a mutation is a permanent change of the nucleotide sequence of the genome of an organism, virus, or extra chromosomal DNA, or other genetic elements. Mutations result from damage to DNA, which is not repaired, or to RNA genomes, typically caused by radiations or chemical mutagens. Errors in the process of replication or from the insertion or deletion of segments of DNA by mobile genetic elements. Mutations may or may not produce discernible changes in the observable characteristic or phenotype of an organism. Mutations play a part in both normal and abnormal biological processes, including evolution, cancer, and the development of the immune system, including functional diversity. Mutation can result in several different types of changes in sequences. Mutations in genes can either have no effect, alter the product of a gene, or prevent the gene from functioning properly or completely. Mutations can also occur in non-genic regions. One study on genetic variations between different species of Drosophila suggests that if a mutation change a protein produced by a gene, the result is likely to be harmful, with an estimated 70% of amino acid polymorphism that have damaging effects, and the remainder being either neutral or weakly beneficial. Due to the damaging effects that mutation can have on a genes, organisms have mechanisms such as DNA repair to prevent or correct, revert the mutant sequence back to its original state. Mutations. Next is recombination of chromosome. DNA recombination involves the exchange of genetic material either between multiple chromosomes or between different regions of the same chromosome. This process is generally mediated by homology, that is, homologous region of chromosomes line up in preparation for exchange, and some degree of sequence identity is required. Various cases of non homologous recombination do exist, however. One important instance of recombination in diploid eukaryotic organism is the exchange of genetic information between newly duplicated chromosomes during the process of meiosis. In this instance, The outcome of recombination is to ensure that each gamete includes both maternally and paternally derived genetic information such that the resulting offspring will inherit genes from all four of its grandparents, thereby acquiring a maximum amount of genetic diversity. Recombination is also used in DNA repair, particularly in the repair of double strands breaks, as well as during DNA replication to assist in filling gaps and preventing stalling of the replication fork. In these cases, a sister chromatid serves as the donor of missing material, recombination followed by DNA synthesis. Next, we will learn about the polymorphism. Polymorphism in biology occurs when two or more clearly different phenotypes exist in same population of a species. In other words, the occurrence of more than one form or morph. In order to be classified as such, morphs must occupy the same habitat at the same time and belong to a panmitic population, one with random meeting. Polymorphism, as described, where involves morphs of phenotype. The term is also used somewhat differently by molecular biologists to describe certain points mutations in the genotype such as SNPs. Polymorphism is common in nature. It is related to biodiversity, genetic variation, and adaptation. It usually functions to retain variety of form in a population living in a varied environment. The most common example is sexual dimorphism, which occurs in many organisms. Other examples are mimetic forms of butterflies and human hemoglobin and blood types. According to the theory of evolution, polymorphism results from evolutionary processes, as does any aspect of a species. It is heritable and is modified by natural selection. In polymorphism, an individual's genetic makeup allows for different morphs, and the switch mechanism that determines which morph is shown in environmental. In genetic morphism, the genetic makeup determines the morph. Ants exhibit both types in a single population. Now, let us learn in detail about the mutations. Mutation is change of nucleotide sequence of the DNA or change in the structure of chromosome. Mutation is basically caused by mutagenes, which are the mistakes in the base sequence of DNA. Here we can see that in normal DNA sequence, thymine always pairs with adenine, but when it is wrongly base paired with cytosine, sequence changes and thus mutations occurs. Now let us see the causes of mutation. There are basically four main causes of mutation: spontaneous mutation due to molecular decay, Mutations due to error-prone replication bypass of naturally occurring DNA damage called error-prone or translations synthesis. Third is errors introduced during DNA repair. And the last one, induced mutation caused by mutagenes. Scientists may also deliberately introduce mutant sequence through DNA manipulations for the sake of scientific experimentation. Now let us learn about these in detail. First is spontaneous mutation. Spontaneous mutation on the molecular level can be caused by first is tautomerism. A base is changed by the repositioning of a hydrogen atom, altering the hydrogen bonding pattern of that base, resulting in incorrect base pairing during replication. Second is deprionation, that is, the loss of purine base A or G to form epipuritic site, that is, AP site. Third is deamination. Hydrolysis changes a normal base to an atypical base containing a keto group in place of the original amine group. Example includes the change of C with U and A with HX, that is hypozetine, which can be corrected by DNA repair mechanism, and 5-MEC, that is 5-methylcytosine with T, which is likely to be detected as a mutation because thymine is a normal DNA base. Next is slipped strand mispairing, denaturation of the new strand from the template during replication, followed by renarration in different spot. This can lead to insertion or deletion. Error-prone replication bypass. There is increasing evidence that the majority of spontaneously arising mutations are due to the error-prone replication or translational synthesis past a DNA damage in the template strand. As described in the article, DNA damage, naturally occurring DNA damage, arises about 60,000 to 100 times per day per mammalian cell. In mice, the majority of mutations are caused by translational synthesis. Likewise, in yeast, Kunis et al. Found that more than 60% of the spontaneous single base pair substitutions and deletions were caused by translational synthesis. Next is errors introduced during DNA repair. Although naturally occurring double strand breaks occur at a relatively low frequency in DNA, their repair often causes mutation. Non-homologous and joining is a major pathway for repairing double-stranded breaks. NHEJ involves removal of a new nucleotides to allow somewhat inaccurate alignment of the two ends for rejoining, followed by addition of nucleotides to fill in gaps. As a consequence, NHEJ often introduces mutations. Let us understand about induced mutations. Induced mutations on the molecular level can be caused by chemicals such as hydroxylamine base analogues Alkylating agents, example N-ethyl and nitrosourea, these agents can mutate both replicating and non-replicating DNA. In contrast, a base analog can mutate the DNA only when the analog is incorporated in replicating the DNA. Each of these classes of chemical mutagens has certain effects that then lead to transitions, transversions, or deletions. Agents that form DNA adducts, example, oscillatoxin A metaboloids, DNA intercalating agents, example, ethium bromide, DNA cross-linkers, oxidative damage, nitrous acid converts amine groups on A and C2 diisogroups, altering their hydrogen bonding patterns, which leads to incorrect base pairing during replication. Next is radiations. Ultraviolet radiations, that is, non ionizing radiation. Two nucleotide bases in DNA, that is, cytosine and thymine, are most vulnerable to radiations that can change their properties. Ultraviolet light can induce adjacent pyrimidine bases in a DNA strand to become covalently joined as a pyrimidine dimer. Ultraviolet radiation, in particular longer wave, can also cause oxidative damage to DNA. Now let us learn about the classification of mutation. The sequence of a gene can be altered in a number of ways. Gene mutations have varying effects on health depending on where they occur and whether they alter the functioning of essential proteins. Mutations in the structure of a genes can be classified as small scale mutations, such as those affecting a small gene in one or a few nucleotides, include point mutation, often caused by chemicals or malfunctioning of DNA replication, exchange a single nucleotide for another. These changes are classified as transitions or transversions, most common in the transaction that exchanges a purine for a purine or a pyrimidine for pyrimidine. A transitional can be caused by nitrous acid base mispairing or mutagenic base analogues such as 5-bromo 2-dioxyuridine, less common in a transversion which exchanges a purine for a pyrimidine or a pyrimidine for a purine. An example of a transversion is the conversion of adenine into a cytosine. A point mutation can be reversed by another point mutation in which the nucleotide is changed back to its original state, that is, true reversion, or by second site reversion, a complementary mutation elsewhere that results in regained gene functionality. Point mutations that occur within the protein coding region of a gene may be classified into three kinds depending upon what the RNS codon codes for. First is silent mutations, which code for the same or a sufficiently similar amino acid. Second is missense mutation, which codes for different amino acids. And the third is nonsense mutations, which code for a stop and cantonate the protein. Insertions add one or more extra nucleotides into the DNA. They are usually caused by transposable elements or errors during replication of replicating elements. Example: 80 repeats insertion in the coding region of a gene may alter splicing of mRNA. Slice site mutation or cause a shift in the reading frame, known to be frame mutation, both of which can significantly alter the gene product. Insertion can be reversed by excision of the transposable element. Deletions remove one or more nucleotides from the DNA. Like insertions, these mutations can alter the reading frame of the gene. In general, they can irreversibly, though exactly the same sequence might in the theory be restored by an insertion. Transposable elements available to revert a very short deletion in any location either are highly unlikely to exist or do not exist at all. Large-scale mutations in chromosomal structure includes amplification or DNA duplication leading to multiple copies of all chromosomal regions, increasing the doses of the gene located within them. Deletion of large chromosomal regions leading to loss of the genes within those regions. Mutation, whose effect is to juxtapose previously separated pieces of DNA, potentially bearing together separate genes to form functionally distinct fusion genes. These includes chromosomal translocation, that is, interchange of genetic parts from non homologous chromosomes. Second, interstitial deletions, an intrachromosomal deletion that removes a segment of DNA from a single chromosome, thereby opposing previously distinct genes. For example, cells isolated from a human astrocytoma, a type of brain tumor, were found to have a chromosomal deletion, removing sequences between the fused in glioblastoma gene and receptor tyrosine kinase ROS, producing a fusion protein. The abnormal fusion protein has consequently active kinase activity that causes oncogenic transformation, that is, a transformation from normal cells to cancerous cells. Chromosomal invergence, reversing the orientation of a chromosomal segment. Next is the loss of heterozygosity, loss of one allele either by a deletion or a recombination event in an organism that previously had two different alleles. Now let us learn about the chromosomal mutations. We should be aware about chromosomal mutation. Sometimes mutation leads to some disease which appear phenotypically. Chromosomal mutation has an extra chromosome. Sometimes during the first or second stages of meiosis, chromosomes fail to separate, which means either the egg or sperm are missing a chromosome. If fertilized, the others will have an extra chromosome, that is, will have three copies, a condition known as trisomy. Examples are Down syndrome, Klinefelter syndrome. In these examples, we can see that in Down syndrome, chromosome number 21 has 3 copies of chromosome, which is abnormal. Let us know about the mutation rate. Mutation rate differ for different genes, which depends upon the size and the sequence. Determination of mutation rate is done by following estimates of unplanned mutation rate may be derived by observing the mutant traits.
Let us now recapitulate what we have learnt in this module. Human genetic variation is the scientific study of inherited human variation. Genetic variation takes place equally in and amongst the population. It is important to study genetic variation to better understand ourselves as a species, and it also provides genetic material for natural selection. Genetic variation occurs due to the differences in the sequences of genes. No two individuals, except for identical twins, are genetically identical. Variation can be seen at every genetic level in DNA, in gene, in chromosome, and in enzymes. The major causes of genetic variation are the changes that occur at the DNA level spontaneously or by the use of chemicals. Such changes alter the sequence of nucleotides. These are known as mutations. Recombination of chromosomes. The genetic rearrangements can affect several genes or large areas of chromosome. Polymorphisms. The differences which occur in individual DNA but are not mutations. Single nucleotide polymorphisms are the most common. Mutation is change of nucleotide sequence of the DNA or change in the structure of chromosome. The basic causes of mutation are spontaneous mutation, error-prone replication bypass, errors introduced during DNA repair, and induced mutation. Mutations can be classified as follows. Small scale mutation like point mutation, for example transition, transversion, insertion and deletion. Large scale mutations like amplification, chromosomal translocations and chromosomal inversions. Chromosomal mutation has an extra chromosome. Sometimes during the first or second stages of meiosis, chromosomes fail to separate, which means either the egg or the sperm will not have a chromosome. If fertilized, others will have an extra chromosome having 3 copies, a condition known as trisomy. Mutation rate differs for different genes, which depends upon size and sequence. Estimates of spontaneous mutation rate can be derived from observation of dominant traits. For autosomal genes, mutation rate is equal to number of cases upon twice the number of individuals.